Hello, student of class 7. Let's continue our discussion. We were discussing about our alimentary canal. We start from mouth and goes up to the anus. And we also discussed that the process of taking food in mouth and putting inside is called ingestion. And we discussed about our teeth, different types of teeth, and our tongue. Now we're going to discuss mouth or buccal cavity. So this is very important, the process of digestion. The process of digestion starts in the mouth, or we can say the buccal cavity. Here the food is ingested and is chewed by the teeth. The chewing of the food is called mastication. Remember, student, chewing of the food is called the mastication. It breaks down the food into smaller pieces for easier digestion. And saliva is digestive juice which is secreted by the salivary gland present at the back of the throat and under the tongue. The digestive juices present in the saliva convert complex carbohydrate such as starch present in the food into simple sugar molecules. So we can say in our mouth, but partially the carbohydrate molecules broken down and convert into simple sugar, and the process of digestion begin in the mouth. We get a sweet taste when we chew rice or bread for some time because of the action of saliva on starch. So here to mention that on our saliva one enzyme is present. This is called salivary amylase. When this enzyme acts the food, we can detect the taste from our taste bud which is present on our tongue, different position. Once the food is chewed, the tongue rolls it into a ball and called bolus and pushes it to the back of the mouth to be swallowed. Here we are going to discuss an activity where we can find what will happen if saliva mix with the food. In our previous slide, I have already discussed with you that in our saliva some enzyme is present, and the enzyme name is salivary amylase, which when act on the food, then we can feel the taste. Now we are going to study the effect of saliva on food, and to perform this activity we require to manage two test tube, dropper, boiled rice and iodine solution. Procedure: take two test tube and label them as A and B. Procedure number 2: put 1 teaspoon of full boiled rice in test tube A. Put 1 teaspoon full of chewed boiled rice into B. So here you have to manage two test tube and label them as A and B, and one test tube you put only boiled rice, and in another test tube you put chewed boiled rice. After chewing or partially chewing the rice, you have to put in the test tube B. Now you add some water to both the test tube, and lastly you pour 2 to 3 drops of iodine solution to both the test tube and observe what happens. You will see that a blue-black color appears in test tube A, while no change in color is observed in test tube B. Remember, student, in test tube B what you put? Chewed boiled rice, and in test tube A simply boiled rice. So you can see in test tube A the color appear blue-black, but in test tube B color is not changing. Why? Because the blue color indicates that the presence of stars in test tube A, and as that stars is broken down by the action of saliva in test tube B gives no blue black color with iodine solution. So we can understand that if stars chewed by teeth and mix with the saliva, then here the color is not going to change because stars already broken in test tube B, but in test tube A, since we have not chewed, the star has not broken, so star is present, and if anything we can check star is present or not, so we always put iodine solution. If iodine solution change blue black color, that means stars is present. It means that in test tube A stars is present, in test tube B stars has broken down after chewing.